Um, Hi, my name is Ashwini Patil. I'm the CEO of Combinatics Japan, and today I'm going to talk about an introduction of single-cell RNA-seq data analysis and methods for cell type annotation, focusing on our cell type annotation database and service CellKb. So this is the outline for my talk. I'll spend a couple of minutes on basic, very basic pipeline in R and Python using Surat and ScanPy for clustering data, single cell data, and identifying cluster specific gene lists, since this is what is done, is what is needed before you move to cell type annotation. Then I will talk about the current methods that are available for cell type annotation and what are the challenges that are faced while using those methods. And then introduce CellKb, the knowledge base, the data collection method, the annotation method that we use. And then finally, I will give a demo of the CellKb web interface that you can use for cell type annotation of single cell and spatial RNA-seq data, as well as bulk RNA-seq data. So a little bit about my background. So I have a PhD in bioinformatics, and before starting Combinatics in 2020, I was an assistant professor and lecturer at the University of Tokyo for over 10 years. And since 2020, I have been developing CellKb, and we have been continuously releasing it for the last 5 years. It has grown a lot, and I will talk about the contents of how they have grown and so forth. So let me go ahead and start with the actual talk. And thank you, Fatih, for giving this opportunity to speak to the users of the immunophenotyping core. I hope they will be able to use CellKb and find it useful in their research. So the first step for any single cell RNA-seq data analysis is basically obtaining the UMI counts, right? So once we sequence the data, run FastQ, map the data and generate the counts using Cell Ranger if you are doing single cell, or spatial RNA-seq data, if Space Ranger if you're running spatial RNA-seq. You generate the counts, and the counts are usually obtained in the form of an H5 file, or you get these three files from Cell Ranger: the matrix, the list of cells, and the list of genes. And what you have to do at the end of this process is basically take these and go through a standard pipeline, at the end of which you get clusters or individual cells and you want to annotate them. So there are currently three main ways in which you can do this. One is using the Loop browser or a third-party tool that you can use for performing the data analysis without programming. The more common ways are using R or Python to actually programmatically do the initial data analysis followed by the annotation. Okay? So this is what I will focus on for the next couple of slides. So here is the standard pipeline that we follow. And maybe most of you are already using this and are aware of this. So I will just go over it quickly. So the first thing is to load the count matrix that you are going to get from Cell Ranger. You filter the low quality cells, normalize and scale the data, perform dimensionality reduction using principal component analysis, generate the UMAP for plotting. This is followed by clustering the data and identifying cluster-specific genes. And this is actually the main step that allows you to then do cell type annotation. Okay, so the reason these are actually not bottlenecks, these are very easy to do. The only part that we need to decide is how we want to filter the cells and how we want to cluster them. So those are the two important points that we need to take care of before we come to the cell type annotation. And then once you have the annotation, you go to the downstream analysis. It is also possible to actually skip the clustering, and you can directly from dimensionality reduction, you can actually go to cell type annotation. Though it is not usually done. And in fact, a lot of pipelines will allow you to do both. So you can annotate cells as well as annotate clusters. And in reality, actually, this pipeline is never one way. So you annotate cells, and then after you go through one round of annotation, you will usually recluster your data at a higher or lower resolution and then re-annotate this data, re-annotate your cells. So you go through multiple cycles until you reach a final annotation that is satisfactory. And then you follow the downstream analysis. And that is the reason actually this is a big bottleneck, because you have to go through multiple rounds of doing this, and for each round you need different reference maybe to do this annotation. Okay, so I just want to quickly go over this, and I'm sure a lot of you already know this, but so there are two main libraries that you can use to run any single cell pipeline. One is the R library Surat, and all the details are actually on this website, and they have excellent tutorial that you can follow. Even if you don't have a lot of experience working with R or no experience working with R, you can actually follow their code line by line and analyze your data. Okay, so I'm just going to go, this is the code for Surat. And the reason I'm showing this is because I want to set up the creation of cluster specific gene lists. Okay, so this is one of the ways that you can generate cluster specific gene lists for annotation. So the first step, and these commands are taken exactly from the Surat tutorials from their website. So you can actually copy them, and then these slides will also be shared along with the comments over here. So you can copy it from here as well. So the first step is loading the data, and there are specific functions to load the data, followed by creating the Surat object. The next step is filtering the data. So in this case, this is an important step where you want to decide what is the amount of, how many cells do you want to keep in your data set based on what is the number of genes present in each cell, and how many genes are expressed in how many cells. Okay? So also, what is the mitochondrial content in each cell? So based on these three things, you can actually decide the number of cells that you want to keep in your analysis. These numbers are standard numbers, and if you generate this violin plot, you can actually see the distribution of these three counts. So you can see the distribution of the number of genes, the number of UMIs per cell, and the mitochondrial content per cell. So based on these distributions, you can decide the cutoffs. Your cutoff could be 500 and 10,000. It depends completely on your data. The next step is normalization. Finding the variable genes, the genes which are show the greatest differential expression across cells, scaling the data, and then dimensionality reduction. This is standard. You can decide to change the number of features that you want to look at, or the genes. You can make them 3,000 or 4,000, depending on how many genes you want to keep in your final analysis. This is followed by clustering. For clustering, you have to decide how many principal components you are going to take from your dimensionality reduction. And that is actually decided by this. You can tell that from the elbow plot. So wherever this plot sort of levels off, those are the number of principal components that you can pick. Usually 20 to 30 principal components is a good number. Also here, the important thing is the resolution for clustering. And this is something that this factor and the filtering, these are the two things that you will probably have to tweak or change with multiple rounds of annotation and clustering. Okay, so initially you set the resolution at 0.5 and you will get some number of clusters. You can annotate them. Once you annotate them, you realize that maybe there is a cluster that is too large and you want to split it into multiple clusters. Then you can either separate it and recluster, or you can increase this resolution and get a greater number of clusters. Okay? And so those clusters can then be re-annotated. So the resolution and the filtering criteria are actually the important things that will affect greatly the annotation that you finally get for your data. Okay? And then once you have the initial clusters, you can identify the cluster-specific gene lists using this find all marker function. And you can keep so the p-value is calculated by default using the Wilcoxon rank sum test. And you can have different thresholds for log4 change or p-value, depending on how many differentially expressed genes you can get per cluster of cells. Okay. And then once you write this into, and then of course write it to a file, and then this is what you can use to now annotate your data, either based on clusters manually or in automatic, using automatic methods. So using these at 4 sort of steps, it's straightforward to come up with the list of genes, which you can now use for annotation. And the Surat tutorial is actually very good. So please try it. If you haven't tried it yet, it will give you a lot of flexibility in terms of identifying, what is the resolution you want for your clustering and what are the filtering criteria. And the same thing also goes for ScanPy. It's basically the exact same pipeline. You can install it. They also have their tutorial, and you basically have you can have the same 4 steps with similar data, though here they are also checking for ribosomal genes and hemoglobin genes, but you don't have to do that. But it's basically the same workflow, and you ultimately end up with this list of genes for the clusters, which you can use for annotation. Okay. So once you have the initial list of genes that you want to use for annotation, you basically can use either one or both of these methods, right? So you initially have your data, and this data specifically doesn't need clustering, automatic annotation. So you can perform automatic annotation using one of the many methods, and some of them I will go over in the next slides. And then this is followed by manual annotation, and this is usually based on the clusters. And then finally verification or statistical support, independent data integration, and so on and so forth, right? So in a lot of cases, what happens is the automatic annotation is not good enough, or it is not clean enough. So usually what we do is we do the automatic annotation and then follow it up with a manual annotation that is based on the clusters to get a cleaner UMAP or a cleaner separation between the cell types. And this manual component actually takes up a lot of time. Okay. So I will go over each of the 2 methods a little bit. So for manual annotation, what is essentially done is you find the canonical markers of the cell types that you think are present in the dataset or are present in the dataset. So if you are looking at PBMCs, then you roughly know what kind of cell types you are looking for. So you can find their marker genes either from databases like CellMarker or PangLauDB, or you can collect them manually from literature and then plot them on the UMAP and see which clusters are expressing those genes. And based on this expression pattern, then you can assign an annotation to that particular cluster. So this is, of course, time intensive and it is not scalable, but it is, even after automatic annotation, this is actually a good way to check your annotations. So manual confirmation of annotations is essential even after automatic annotation. So it's good to have a list of these canonical markers, which is not always available for all cell types. Okay? But I will go over how CellKb helps with that as well. The other method which is also used is the automatic. These are the automatic methods. And the automatic methods of cell type annotation are divided into three main categories. One is the classification or reference mapping based methods. So in this case, what happens is a single data set or data from multiple publications is integrated to create an integrated reference, which is used to train a model that can then be used to assign cell types to single cell data. And in this case, the cell types are assigned to individual cells and not to clusters. So here we are talking about annotating each cell in the single cell data set separately. And some of the very popular methods are actually cell typist, which you can run in Python along with the ScanPy pipeline. Singular, which you can run in R. Azimuth, I think it runs in R. So you have a lot of these methods and they already have some pre-trained models that you can call and use, and they are quite fast. So the only issue with these methods is that the models are not updated and the references are not comprehensive enough. Okay? The newer methods are AI methods, which are based on foundation models. And these are models that have been trained on massive single cell data sources like cell by gene, CellX gene, and also custom databases which users create by integrating a large number of data sets from GEO, for example. So some of the commonly used methods are SCGPT, SC similarity, gene former. All these run in Python. So you need to actually install them and install several libraries to run them. And you also need an access to GPU because it doesn't work on the normal like a laptop or a local machine. So you need a GPU processing unit. These are also not as frequently updated because it takes a long time to actually train these models on the data. And the final method is the signature matching method. And this is a method which we use in CellKb. This is a rank-based matching method where you match or you compare cell-specific or cluster-specific genes to cell type signatures from reference publications. So it's actually very fast and it doesn't require a lot of computational power. The only thing it needs is a really good reference database because you want to have a comprehensive set of cell type signatures that you will compare your cells or clusters to. So that is what we have done in CellKb. Okay? And I have a lot of details about each of these methods and a comparison on the CellKb blog, for which I will share a link in one of the later slides, along with um, with results of actual runs of these tools. So with all these tools, the main problem is that it's very difficult to find the right reference. So cell type annotation is actually based on finding a reference that contains the cells of types that you are looking for, right? And if you look at how, and most of these references are single cell references, so single cell data, other than, for example, immune cells for which you have an excellent bulk reference like InGen. Other than that, for all other cell types, you need to look for a single cell data set that is going to have cell types that match your experiment. And over the years, you can see that the number of publications that have single cell or keyword, scRNA-seq keyword in their abstract has increased exponentially. So this is actually data from PubMed of the number of publications that have single cell related terms in their abstract and text. So it's very difficult to actually find the right reference. Even if you find multiple references that will work for your annotation, there is no standardization in the cell type terminology that is used. So some of them might be, they might call CD8 plus T cells or CD8 memory T cells or CD8 um, effector memory T cells. So they are not only non-standardized, but they are also at different levels of granularity. Also, not all references contain the complete cell type information for a particular tissue. So if you are looking for cell types in spleen, one dataset might contain only the immune cells, another dataset might contain the non-immune cells. So you need to integrate basically multiple references to get something that is comprehensive. And this integration of multiple data sets is a non-trivial task. So this is one challenge. The second challenge is of annotating diverse cell types. So this is the number of cells that were identified in studies that we have in CellKb, in the CellKb database that we have collected. And this is a logarithmic plot. So you can see that, in 2000, so this is too early actually, very few cells, but recent data, you have an integrated data set of 21 million cells. Now, if you have a data set of 21 million cells, in order to annotate those cells, you are going to need a lot of references. And not all of them will be at granularity that you want. So a lot of references, if you look at them, you will get annotations which are like hepatocyte 1, hepatocyte 2, macrophage 1, macrophage 2, macrophage 3. And you don't know what type of macrophages they are actually, or hepatocytes they are. But you want to know whether it is a um, periportal hepatocyte or a mid-zonal hepatocyte. So the other thing that happens is in automated methods, the predictions are all black box. So you don't know where the prediction is coming from, which reference is used for a particular cell type prediction. Also, a lot of the automated methods for which we have existing models are for human and mouse. And so a lot of the model organisms are actually, you have to annotate them manually. And of course, the databases that are, or the models underlying several methods are infrequently updated, which means that um, if you have a cell type that has been identified in 2024, you won't be able to catch it because your reference contains data from 2021. So that actually affects the annotation of diverse cell types because it limits the number of cell types that you are going to find in your reference. So for these two challenges, to address them and make it easier to annotate cell types, basically, we have developed CellKb. And CellKb is a database of cell type signatures, which we have collected manually from literature. And it is supported with a method for cell type prediction that is fast and accurate. Um, so currently the recent CellKb release contains 121,000 cell type signatures across 6,000 cell types from 13 species. And these have been taken from 1,600 publications, which we have picked for high quality. Okay? So I will go over how we selected them. But this is how CellKb, the data in CellKb has increased, and this is the number of cell type signatures, where a cell type signature is a list of genes that is upregulated in that particular cell type in that experiment compared to all other cell types in that experiment. So that is the definition of a cell type that we are using here. So initially we started, the first release was in April 2020, and now actually in the next 2 weeks, we will have our next release on the 1st of August. So, and that will increase the number of cell types to maybe 140,000, around that count. So we have increased, the data has increased a lot, and we are constantly getting new data from literature and adding new signatures. So this is the breakup of the data. Of course, most of the data is human and mouse because it is a reflection of what is available in literature. But we also have a lot of representation of cell types for other model organisms like zebrafish, fly, monkey, Arabidopsis. Um, and of course, we also have information about diseases because we do collect information about tissue and disease conditions. So and we add species based on user requests. So if there is a species that you are interested in and that is not here, we can add it for you. And currently, our user base consists of pharmaceutical companies, biotech companies, bioinformatics companies, in a lot of university and research institution cores, individual research labs, and cancer hospitals who use CellKb for annotating single cell and spatial RNA-seq data, and also bulk RNA-seq data actually. So, um, okay. So this is the CellKb knowledge base. This is how we collect the data for CellKb. This is how we collect the cell type signatures. So initially, we take all the publications that have potentially have cell type signatures in them. And these could be bulk or single cell. Mostly they are single cell. So far we have looked at 120,000 papers. And these are, they initially go through an automatic filtering process for which we have a customized tool that we use for automate filtering or identifying papers that have high quality cell type signatures. Once we filter it down to about 20,000 or so publications, we look at we look at these manually. Actually, we have looked at them over the last 5. So this is data over the last 5 years. We looked at them manually to see excuse me, if the cell type signature is of good quality, if the data is of good quality, if the data is publicly available, and so on. And then finally, we have arrived at the current number of publications that have been accepted. Now, for each of these publications, which we have selected using a very stringent number of stringent criteria. we standardize the cell type, disease, and tissue information for each of these cell type signatures from these papers using cell ontology, disease ontology, and Uberon ontology, so that the data is harmonized for rapid searching. We also collect associated values and sample conditions which are assigned by the authors. So in many cases, the data, the signatures are collected from the supplementary materials of the papers, or if they are not present in the supplementary materials, but the authors have published or provided access to their raw or processed data, then we will take those and we will generate signatures for cell types that have been annotated by the authors in the paper. So we will generate those cell types based on the author information. All signatures that we collect are then scored for reliability. And finally, we also collect the canonical markers in each paper that the authors use to assign a particular gene list as a cell type. So we collect the cell type and the canonical markers along with any values, conditions, disease information, treatment information, experiment information, sample information. And then all this information goes into creating the CellKb knowledge base. And this data is updated every 4 months, along with recalculating the reliability scores every single time. Okay. The cell type prediction method is basically a rank-based method. So what happens at the end of this process is we get a database of gene lists. So each gene list is a cell type signature, and with that signature, we have an associated cell type, a tissue, a disease. And now what we want to do is when the user comes with a gene list, either a gene list of a cell or a gene list of a cluster, we want to compare the user's gene list with all the cell type signatures that we have in CellKb. And so the input is actually a ranked gene list. And the ranking of this gene list, the cluster specific gene list, is done using log-fold change values of those genes. Okay. So the method is comparing the input with every cell type signature from the database using a rank-based method. And the rank-based method considers the ranks of the genes in the query list, the cell type signatures in the database, rank differences, the number of overlapping genes, and so on. And this method is actually, the algorithm is specifically developed for CellKb to work with CellKb data, and it works with gene lists of unequal lengths. So matching gene lists of unequal lengths. It is a fast and scalable method, so you can annotate hundreds of clusters within minutes by comparing them with hundreds of thousands of cell type signatures. And we have evaluated this against ImGen, and using this method, the hematopoietic marker gene sets for mouse we were able to assign the exact match, a parent match, or a sibling match, that means related match, in 94% of the cases. And this number actually, it was much lower in 2020 in this paper, but it is increasing as the database increases. Right? And based on this matching, then we get a list of 100 cell type signatures that match the query or the gene list that you are going to give. And based on that, we predict broad and granular cell types for each cluster or each cell. Okay. Usually we give the top 3 predicted cell types with canonical markers for each cell type, and these can then be used for manual confirmation of the prediction and a prediction score. We also statistical significance is calculated for each match, and we provide all the reference publications for the top 25 matches. So we give the paper to which basically your cluster is matching, from which the cell type signature was taken. Right. So you have a link. It's not a black box. You get a link to the paper to which from which the prediction was made. And there is a heat map of similarity between query and top 100. And I will show that a little bit later. Just see. Yeah. So to address the two challenges, right? First is finding a suitable reference. So this actually you don't need to do with CellKb because we have already generated a reference um, using high quality signatures. So we have a large number of cell types, tissues, diseases, and species available in the single curated resource. Multiple publications can be selected as reference without the need for integration. So you can actually select hundreds of papers to compare your cells or clusters with without integrating because we are going to check against cell type signatures from each paper. You have standardized annotations and regular updates. And for annotating diverse cell types, because we have so many publications, we can actually predict uh, high level of granularity for the annotations because a lot of papers do provide good granular annotations, though not all, right? So based on that, we can predict the granular annotations. Since there is no reference aggregation, you get a reference publication for your prediction. Canonical markers are provided for confirmation. And then you can actually also search across species. So if you have an experiment, your single cell experiment is on rat, then there aren't enough references in literature actually for rat cell types. So you can compare your rat data with mouse cell types and you can annotate across species for other model or non-model organism. Or if you have a type of fish that you are studying, then you can compare it with zebrafish, for example. So this is a quick comparison table. And if you look at this link or go to the CellKb website and look at the blog, this gives a lot of details about how this table was made based on what data it was made. But in short, this is the method that is used for, so you have manual, then you have the classification based cell typist and singular, and then you have the AI based methods and CellKb, which is rank based. So manual, of course, doesn't need coding. CellKb has a web interface, so you don't need to code for that. Ease of use, these are moderately easy to use. Actually, if you know how to code, they are not so hard to do. And you can definitely, I would recommend using multiple methods of cell type annotation for any data set. So SLKB is easy to use because you have to just upload your gene lists. Almost all of them require a reference that you need to find. So you have to look for a reference to run these methods. Whereas for these two, the reference is inbuilt. You don't need a specific reference, so you can select one based on filtering criteria. Um, Annotating, so manually, you cannot actually annotate single cells manually. You can annotate clusters. The other automated methods are mostly annotating of single cells. And CellKb, you can annotate clusters and cells. This is the computing resource requirement of obviously the AI methods require more computational resources. Most of them are fast, though, manual is of course slow. But if you have the canonical markers already available to you, then it can be quite fast. Species, this you basically pick the species that you want, right? Most of the data is human and mouse, so that limits it a lot. Standardized ontologies are actually available for the AI methods because they are based on cell by gene, which uses cell ontology. And then actually none of these are regularly updated. So that is a big issue, and that is one of the things that we are trying to address. Okay. So the functionality that is available to you through the core is cluster annotation, single cell annotation, spatial RNA-seq deconvolution, so spot deconvolution, and bulk RNA-seq cell type enrichment. CellKb also has also provides API access. So if you have a pipeline and you want to integrate CellKb functionality or annotation into your pipeline, you can do that with API access. And we have a lot of users who actually do this. Um, and we also have consensus signature generation. Because we have a huge data set, we can generate consensus signatures and cell type specificity scores for genes for different cell types. So you can compare the signatures, for example, across diseases, between disease and normal, and so on. And this requires a separate license, so it is not available through the core. So I was actually going to cover this in slides, the functionality, but I think I will go straight to the demo and show you how to use CellKb. So this is the CellKb website, and on this website you will find the resources and the blogs which give you how to use CellKb, and this is the blog about comparison. You can also see the citations and what are the other papers that I have been using CellKb in annotation. Okay. If you go to the login page, uh, you request Fati for user ID and password. And then once you have that, you can actually log into CellKb from here. Okay. And I have already logged in. So if you look at this, there are two main menus. One is just exploring the data, looking at the data, and this is the main menu. Here you can annotate clusters, look at the clusters that you have previously annotated. You can annotate single cell data by uploading raw UMI counts, spatial data by uploading Visium UMI counts from Visium data, bulk RNA-seq data, and enrichment analysis also actually you have access to. This is for identifying cell types in unranked gene lists. Okay. So first, the cluster-specific search is actually the most frequently used in CellKb. And in this, you can take the output of Surat. So this is output from the Surat pipeline of cluster-specific genes. And here I have 6 clusters which I want to annotate. So if you give the cluster ID, it will automatically annotate all the clusters. You can over here, you can annotate across species. So if you have, you can pick human versus mouse, for example, if you don't have enough data for your species. Um, we also have bulk data, but it is not as large as the single cell data, but you can definitely select bulk data. So we have data from ImGen, we have data from Human Protein Atlas, MSIC-DB. So you can actually use bulk data for annotating clusters which are tricky to annotate. You can specify a tissue that you want to look at, and this count is the number of cell type signatures that are available for this tissue. So you can include and exclude tissues, you can include and exclude cell types. Okay. I don't want to include anything. You can also include diseases. So if you want to look at only specific disease-related signatures, or create a reference that consists only of disease-related signatures, you can do that. You can also select papers up to a certain time. Okay? And actually in CellKb, there is a condition type that you can select. So when we collect signatures in CellKb, we collect 4 types of signatures. signatures that are taken from normal cell types or normal samples, signatures collected from single cell data that is a mixture of normal and disease or treated samples, and signatures that are taken from only disease or treated samples, which is here. And finally, signatures which are relative signatures. So those that are taken in experiments where you are comparing a disease or a treatment sample with a control sample. So if you are doing a relative comparison, then you need to pick this condition and actually remove these conditions. But in most cases, you will not be using this. You only need these conditions. Okay? And then if you want to only use normal samples, you remove these two conditions and then you just pick control. Okay? Then you search, and um, if you have, so you can enter a gene list which is ranked. You don't need to enter these values actually. CellKb doesn't need these values as long as your gene list is ranked. If you don't rank it, then and you enter the values, then what CellKb will do is it will take um, genes which are below this p-value threshold and above and are upregulated basically. And then the genes are sorted by log-fold change. So it will rank those genes based using those criteria. It will also, since it knows what data to expect from Seurat and SCIC, it will guess. But if it doesn't guess correctly, you can actually change this field to set it to the right field. Okay. And the input gene list can be an ensemble ID or it can be the gene name. So now this is actually running. So this is live. And so for what happens is this is the result. So for every cluster, we have 6 clusters. It has made a cell type prediction. Okay. And cell type predictions with a score greater than 0.5 are considered reliable or are considered not exactly reliable, but that is probably the cell type that this cluster is. Sometimes you will get scores which are equally divided. So we predict up to 3 cell types. And so the 3 cell types get scores which add up to 1. So sometimes what can happen is you will get 3 predictions which have very close scores like 0 0.3, 0 0.3, 0 0.3. And then you have to make a call of which cell type this cluster could be. But in many cases, based on this information, you can actually figure out what the cell type is, if not directly based on this. Okay. Now, this relationship is showing granular versus broad cell type. And this relationship is taken from the cell ontology hierarchy. So naive thymus CD4 is a T cell is a child of naive T cell. In the brackets, you can see the canonical markers that have been so far used to identify this cell type in various publications. So you can actually take this and then run it in Seurat or SCIC to check how well these genes are expressed. Okay, and these are the genes that are overlapping with the gene list that we have given for this cluster. The next part of this result is the top 10 hits. So this list is the top 10 cell type signatures in the database that are matching to our cluster. Okay. And for each of these signatures, then we have the tissue from which this cell type was taken, the disease condition, the rank-based score, which is calculated using 3 criteria of comparing the cluster-specific gene list with the gene list that is present in the database. You have the FDR and the number of overlapping genes. So because CellKb compares the cluster-specific gene list with all the cell type signatures in the database, you can actually get up to, if you go to the details, you can get the top 100 hits. So these are the 100 cell types signatures to which our query gene list is matching. And if you look at these, you can see that they are from, it will give you the name of the paper. So it is telling you, so this is actually the list that is given as a sample, which is why it matches so well. But if you look at the others, they are from actually different papers. And you can see the number of overlapping genes, the scores are different. Okay, so each of these is a different paper, a cell type signature that has been taken from that particular paper. And the stars indicate the canonical marker that was used in this particular paper to say that, this gene list that we identify, the authors identified, is naive CD4 plus T cell. Okay. So, and with this information, actually, it's very easy then to say, okay, I know that this is a naive T cell because I am getting so many hits of naive CD4 naive T cell, plus the canonical markers that have been used for naive CD4 naive T cell are very highly ranked in my gene list. So, based on this heat map and this result table. This result table actually gives you the scores, a link to the paper, and then details about the publication of the match. Okay. So if you go here, this is actually CD8. Let's go to CD4. So if you drill down, so this is where we got the initial result. This is the heat map. And then if you drill down, you can see what is the paper, what was the experiment that they performed, how many samples did they have, what are the conditions, what is the cell count, and then what is the reliability. This reliability score is calculated by comparing the signature that we collected from Krishna's cancer cell with all other naive CD4 plus T cells that are in CellKb. Okay. So how well does it match all other CD4 T cells in CellKb is how this reliability score is calculated. And this table shows, this is our query. So we entered this gene list for cluster 1, and it shows the overlapping genes with this paper. So you can actually drill down into which paper it matches to and what is matching in that paper to identify your cell type or to justify, to find the evidence for this prediction. Okay. And you can see here this sample list that I have used. You can actually get very granular annotation. So for example, plasma cell, you get IgA plasma cell. For fibroblasts, it will tell you that this is a cancer-associated fibroblast based on multiple publications. Okay. You can see others. For example, here you have a prediction for a macrophage. And it's possible that this is a mixture of fibroblast and macrophage, but mostly this is going to be a fibroblast because it has a very high score. Okay. And if you don't give any tissue, it will actually also tell you the tissue. In this case, you can see that this is an intestinal cell or a stomach cell. So gastric goblet cell or intestine goblet cell. So you can either select a filter or not select that. It works both ways. But in any case, the number of publications that you are using as reference is 1,686 in this, or whatever is the number of publications for human. And then if you select it to lung, limited to lung, for example, then you would select all the publications that have lung without integrating, but comparing with each signature. And then you can download all this data in TSV format or GCT format. Okay. And in the downloaded data, you will get the broad cell type, the granular cell type, and all these 3 cell types with the scores and the canonical markers. Okay. So the GCT file is the heat map. So this is annotating clusters. You can go back to the history and you can see the clusters that you annotated. You can see this result again. You can search it again with a different filter. And many times what happens is if you are looking at, let's say, 50 clusters and you annotate 50 clusters using CellKb, maybe 45 of them are annotated immediately. Clearly, they have some annotation, but 5 clusters are tricky. You don't know, is it a monocyte or a macrophage, or you don't know exactly whether it's a fibroblast or a pericyte. You could have that kind of situation. In that case, you can actually go back, click on this number here, and then just run that particular cluster again to re-annotate that cluster. Okay. So that is also possible with this interface. And these are deleted within 24 hours, every 24 hours. So you should definitely download all the results that you generate when you run. The next uh, is annotating single cells. And here you can upload directly your H5 file or the matrix, the 10x matrix. Once you upload it, so I've already uploaded it. It takes a little time to upload. You can analyze the sample and you specify the sample that you want to look at, the tissue you want to look at. Do you want to filter the data or not? What are the filtering criteria that you want? And then you can check what is the reference. So in this case, if I look at spleen, then I'm going to have a reference of 25 publications and 120 cell types that I will compare CellKb, that I will compare my data against. And then once I run it, this takes about 5 to 10 minutes to run, so I won't run it now. But you can download this data and it will give you a result that looks something like, let me share my whole screen. That will look like this. So you will get an annotation at 3 levels: broad, subtype, and granular. And then if you zoom on this, you can see in this broad, you have B cells. And then these B cells are actually, you get memory B cell, germinal center B cell. T cells are split into CD4 plus T cell, CD8 T cell. Conventional dendritic cells are then further in the granular annotation split into conventional dendritic cell type 1 and type 2 and so on. So we have 3 levels of annotations, and you can actually also see where each of those annotated cells are. So this annotation is not as clean as cluster annotation, but it does help find small clusters of cells which you would otherwise miss, or which would get included into a single cluster, for example, over here, right? So, and for this also, you can download all the data, download all the scores. And here is, for example, the text result that you will get. So for each cell, you will get broad, subtype, granular, and then the ontologies, the scores for the 3 predicted cell types of each cell. Okay. Similarly, you can also run spatial RNA-seq Visium data. So you can upload the sample for Visium. Here you have read Visium. You can pick a species that you want. Again, once you have uploaded, you can analyze, select the sample, select the tissue. Let's say I have uploaded data that is already normalized and I don't want to normalize it in CellKb. And then I check what is the size of my reference, okay, and then submit. Once you submit, the job will go into a queue and then it will be processed in the order that it was received. And then you can download the result. If you download the result, actually you will see that it contains a lot of text files which um, are the actual results and also the filtering criteria that you have given. And you will also see the annotation. So in this case, again, you have the broad, subtype, and the granular. So for example, you have in this case, you will get glutamatergic neurons for each layer, uh, GABAergic neurons of different types, right? So that is how the annotations will happen per spot. And per spot, we make three predictions for up to three cell types. And the percentage of those cell types is basically normalized so that it comes to 100. So, and this plot shows the one that has the maximum score. And then again, you can see the location of each of those cell types in this plot. And then this data is also available as a table where you have each spot and then the broad granular cell types and the proportions of each cell type. Okay. And then finally, for bulk, this is actually an interface where you can upload bulk RNA-seq data, and it works best. You can upload normalized expression values, but it works best with log fold changes. So if you have bulk RNA-seq samples with log fold changes, you can actually specify different tissues for each of the samples. So here I have uploaded three samples, with log fold change values. And for each of these samples, CellKb can predict the number of the cell types that have been enriched or depleted based on the log fold changes of the genes. So because we have a large database, we have the cell type specificity scores for the genes. And so using those scores and the log fold changes that are given by the user, we are able to predict the enrichment or depletion of cell types. And this is also granular and broad. Excuse me. And you can also look at the table, and all this is downloadable. All this information is actually available on, excuse me, sorry, on our webpage.